Okay, let's keep going. So after you produce sperm and egg, you have fertilization. And because the sperm and egg, each one, those gametes are haploid. They only have 23 chromosomes. And after fertilizations, you have the diploid. You start to have 23 pairs or 46 chromosomes. And because of the random divisions and the crossing over, those gametes, their DNAs are not exactly the same, so which increase the diversity. And that's the sexual reproduction, the purpose to increase the diversity. So when we list, okay, the factors contribute to the variation among offsprings, why our siblings and us, we look different because we have the crossing over. And we also have the random alignments, and we also have the random combination during fertilization. So all this increase the diversity. Okay, so that's the difference between the mitosis and meiosis. So mitosis, the cell divisions happens, and the meiosis, the DNA duplicate and divide twice. And each in the end, we end up with 4 haploid cells. That's the meiosis. So that's the difference between the mitosis and meiosis. Okay, now let's look at the sperm and the egg production. So produce sperm is called the sperm. You have two processes. The first is called the spermatogenesis. Spermatogenesis, it ends up with 4 haploid cells, but actually they are not sperms. They call the spermatid, not sperm yet. So in the spermatogenesis, the focus is the DNA. DNA duplicate and then they divide and then divide again. So you end up with 4 haploid cells, but they are spermatid. They are not sperm yet. The reason is the sperm have to swim through the whole uterus and to the fallopian tube to find the egg. So this kind of shape of the cells, spermatids, they are still not qualified to swim those long distance. So physical maturation is called the spermiogenesis. This one is not the DNA. It's mainly focused on the structure. So they have to gradually develop into those long phalangula, and the nucleus go to the head part, and the cytosol is the body, and these long phalangula. They are able to swim through the whole uterus and to find the egg. So there's the sperm production, includes spermatogenesis. That's the DNA duplicate, divide, and then spermiogenesis. That's the physical maturation. Okay, to produce egg is called oogenesis. And oogenesis is produce egg, very different from sperm production after in the male's testes after puberty. Every day, the sperm is produced. So every day you produce millions of sperm in the male's testes. Female is different. Every month you only have one egg mature. So oogenesis, every month, one egg cells, they start to gradually develop and mature. And you find when they divide to one big one, one small one. And the small one may just disappear. And those big ones, secondary oocyte, they will divide again. So meiosis 1, meiosis 2, and they still divide to one big one, one small one. So you end up with only one egg during the oogenesis. So in females' ovaries, every month you only have one egg mature. And we know during the meiosis, you start to have those chromosomes, random separation, and before that you have the crossing over. In theory, everything run well, but the truth is mistake can happen. So you try to increase the genetic diversity, but the price you pay is sometimes the mistake can happen. So let's look at those mistakes in meiosis. It can happen in chromosome number. It can happen in chromosome structure. So chromosome number is when they divide. you have those non-disjunctions. They suppose divide into two, but they just all go to one side. Those sister chromatid, those homologous chromosomes or sister chromatid, they divide incorrectly. Or in the chromosomes numbers during the crossover, they supposed to switch, but they may not, or a piece left out. So they can have a mistake. And to identify the chromosomes health, we have the karyotype. Karyotype is you line up all the 23 pairs of chromosomes together and you look at those structure. So that's to identify, do you have any missing chromosome or extra chromosome? We do the karyotype. Okay, so let's look at those mistakes. During meiosis, they supposed to divide equally, but you could have those non-disjunction, means they don't divide equally. They just all go to one side. That's non-disjunction happens in meiosis. And then after meiosis 1, you have meiosis 2. Then, okay, well, now the one on the left, you have 3. The one on the right, you only have 1. And they divide, you have the whole chromosome number end up with +1 and -1. And when this kind of sperm usually happen in the sperm meet the egg, and then the chromosome number will be different, won't be correct. And that's the non-disjunction. And non-disjunction is the chromosome number change. And some of them, actually a lot of them, will end up with a very dramatic disaster during the embryo development. Means the embryo won't develop. A lot of cases is this. But some of them, the embryo can survive. And like the Down syndrome. Down syndrome is the extra pair of chromosome number 21. You have extra pair of 21. And Turner syndrome, we're going to look at each one of them. That's you miss one. We call it XO female and Klinefelter syndromes. That's the super male. You have extra pair. So let's look at each one of them. So Down syndrome is when you do the karyotype, you see, okay, all these 23 pairs and this pair, you have extra one. So chromosome number 21, that's the extra, that's the Down syndrome. So these are the characteristic. They have those facial structures, and also they have some affect their IQ and other part of the body will be compromised. And then another one is the Turner syndrome. Turner syndrome is when you look at the karyotype, you find, okay, well, the last pair of chromosomes supposed to be the XY, and you only have one X. So it's missing one. And the Klinefelter syndrome, that's the, you have 2X, 1Y, so extra one pair of the sex chromosome. And these are the symptoms. So, and the last part, well, let's look at the gender development. So the default type, the embryo, the human embryo, we're talking about human, they develop, they need to have, the default type is female. So if nothing happens, the embryo would develop into female. And why some embryo develop into male? Not just because of the Y chromosome, also because on the Y chromosome, you have a gene called SRY gene. So this gene will trigger the chromosome develop into testes. The reason is these genes will make the testosterone release. You know, this is the male sex hormone. So during the embryo development, if you have testosterone, and then it will develop into the male. So these hormones, it will develop into male. If you don't have anything, it will develop into the female. That's the default type. The right side, that's the default type. So default is female. And we know the hormones need to bind with the receptor. When we talk about the membrane proteins, it's the receptor. So SRY genes will trigger the testes develop into release testosterone, and testosterone is supposed to bind with testosterone receptors and trigger the response. However, if the receptor don't respond to the hormone pretty well, it end up like you don't have that hormone. So it's like diabetes. We have diabetes 1. That's because the persons don't have insulin. But we also have diabetes 2. Diabetes 2 is called the insulin insensitivity. The person have insulin. This person usually have more insulin than they need, but end up with their body don't respond to insulin. So the result is the same like diabetes 1. Their blood sugar level could not be controlled. It's because the receptor part, insulin receptor, don't respond well to insulin. And similar here, for some embryo, the body don't respond to testosterone pretty well. So testosterone is supposed to trigger the embryo development to the male after they have testes, but their body don't respond to androgens, the male sex hormones well. So it turned out they still develop into female's body. Jamie Lee Curtis, she's pretty famous because she just got the Oscar. And she looked pretty. She had a very nice body, but actually she's XY. In her body, she has the androgen insensitive syndrome. So that's another things can happen during the embryo development. So all these females, they are XY. They have testes instead of ovaries. So they all have the androgen insensitivity syndrome. Okay, there's something can happen during the embryo development. Okay, let's stop here.